I should say I'm Nancy Kanwisher. I'm a faculty member here. And I'll talk for 20 minutes or so on some general background about the organization of the brain that we've developed over the last 15 years from work with brain imaging that forms the basis for a lot of the stuff we're doing on kids. I've mostly worked on adults, so that's what I'll talk about. And then Danny Dilks over here will talk about some of the questions we're trying to look at with the kids. And then Rebecca will talk about some of her work on probably the coolest part of the brain. There are not very many people here, so feel free to butt in and ask questions. None of this stuff, at least none of the stuff I'll talk about, is that fancy and complicated that there's any excuse for leaving you behind. So if you don't know what I'm talking about, raise your hand or shout. I read an article in the New York Times today about death by PowerPoint. I started thinking, oops, but I'm very much in the habit, so pardon me. Hopefully I won't be too deathly. It was sort of about how deathly military PowerPoints are. So I immediately said to myself, well, that's just military PowerPoint talks. I don't know. Anyway, so this is the team in my lab, and you can see some of the people who are involved in scanning kids here. This is Ale, my lab manager, Kenny Coldewin, who's been very involved in this project, Josh right above, and Sarah, who's right here, and who am I leaving out? Danny in the way back will be talking next. So I'm going to start with the ancient history of the questions we're asking. And this starts way back in the 1700s with the Viennese neuroanatomist, Franz Joseph Gall. And he had two really big ideas. The first was that the brain is the seat of the mind. And the second one is that they're distinct mental faculties or mental abilities, each of which lives in a different part of the brain. So those were two real biggies. He could have made a whole career out of that, and he'd still be famous today for just that. In fact, he should have quit while he was ahead. But he didn't. He really wanted to know what were those mental faculties and where were they in the brain exactly. And back in the 1700s, the methods weren't so great for doing that, so he did the best he could. And he invented the infamous method of phrenology, which you may have heard about, where you feel for bumps on the skull and you try to make the assumption that where there's a particularly big bump, that corresponds to a piece of brain underlying that will be correlated with that person's particular mental abilities. It's an idea. From this method, he came up with 27 different mental faculties. My favorites are amativeness, veneration, filial piety. There are lots of funny ones on here. So people love to diss Gall, even though in many ways, conceptually, he was the founder of our field. One way of looking at it is it was really the right idea and just the wrong method. And we're very privileged to live now where we have the right method to ask this question. And so most of the work that we're doing uses this method here, functional fMRI, or functional MRI. Functional MRI is just like regular old MRI. These machines that probably most of you have seen in hospitals, there are thousands, maybe tens of thousands of these around the world. But there were two key advances that were made by some brilliant physicists. I had nothing to do with these advances. I'm just the happy beneficiary. First, they figured out how to take the pictures really quickly. So it used to be that you'd stick a knee in the scanner and it would take 45 minutes to take maybe 10 pictures of the slices through your knee. These guys figured out how to take those 10 pictures in a second or two, right? So that's one major advance. The second advance is even cooler. They figured out how to take pictures that show you not just the density of tissue, where the bumps and folds and holes and wrinkles are in the brain, but where the activity is. So the basic idea is this. If there's a part of the brain, say right here, that gets very active when a subject does a task, that increases blood flow to that region because neural activity is metabolically expensive, and those neurons need extra blood to supply them with oxygen so that they can fire. And it turns out that actually the blood flow more than compensates for the oxygen use. So there's this weird thing about the fMRI signal that it's backwards. It actually reflects a decrease, not an increase, in deoxyhemoglobin relative to oxyhemoglobin. Doesn't really matter. The point is that oxygenated and deoxygenated hemoglobin are magnetically different, and it's that difference that we pick up with an MRI scanner. So if I lost you in there, doesn't matter. All that matters is neural activity, brighter image on that, brighter patch on that part of the image of the brain. Okay? So this is a truly magic device. It enables us to ask all kinds of interesting questions. So we can return to the kinds of questions Gall was interested in and ask, are there specific bits of brain that do different things? And if so, what are those specific things that they do, and where are those bits in the brain? Okay. So when I first started doing functional MRI, I thought we'd give this a whirl, and I gave myself an easy test by looking for something for which there was really excellent prior reason to think it was in there in the brain, and that's the case of face recognition. And the reason we thought that there'd be a special piece of the brain for face recognition, actually there are many reasons before we did any of this work. One is that we recognize faces in a very different way from how we recognize objects. I'll show you an example of that in a second. Another is that people who had the great misfortune of having damage to the back end of their right hemisphere sometimes had this strange phenomenon where they'd selectively lose their ability to recognize faces. They'd still be able to recognize voices, to recognize objects, to read, to navigate around in the world. They weren't confused about different people. They could tell you everything about a person from their name or their voice. They just couldn't figure out who that was by looking at their face. So it's a very specific disability. So we thought, well, there's lots of reason to think there's a special bit in there for face recognition. Let's see if we can use this machine and find it. Okay? So we did the extremely simple-minded thing of popping subjects in the scanner and showing them pictures of faces and showing them pictures of objects. So this is a 5-minute scan. This is time going this way. So the subject's lying in the scanner. They're looking at images in a screen over their head. And first they stare at a dot for a while. Then they see a bunch of faces come on for a while. Then they stare at a dot. Then they see a bunch of objects, and so forth. Turns out it doesn't matter what the task is. Sometimes we just tell them, hold still, look at the stuff. Sometimes we give them a task to do, like press a button whenever 2 consecutive images are the same. We get more or less the same data in any case, as long as the subject doesn't fall asleep. When they fall asleep, you don't get the same result. So then what we can do is we're scanning their brain with functional MRI the whole time they're looking at this. Once every 2 seconds, we take a whole series of pictures through the brain, so like this, in 2 seconds. They do that all the way through. So then we can take each patch in the brain, and we can say, we can ask statistically whether the MRI signal, remember it's correlated with neural activity, is higher in that little piece of the brain when the subjects were looking at faces than when they were looking at objects. And when you do that, here's me, whoops, here's me doing this task circa, I hate to say, around 1995 or something. This is a slice through my brain like this. A horizontal slice is the back of my head. And so this is, the left and right are flipped here. The radiologists started that tradition. It's very irritating, but it's harder to fight them than to follow. So just think everything's flipped here. Actually, it may not be flipped in your data. Oh well, it's flipped in my data. So what the statistics are telling us here is that little patch there, as well as this other little annex, the neurons were more active when I was looking at faces and objects. Okay? Now, when you see nice colored blobs on the brain like this, which you do in the pages of the New York Times and Newsweek and wherever you look these days, be suspicious. There are lots of ways to cheat and get a nice little blob like that. But we don't cheat, and the way you can tell we're not cheating is I'll show you my actual data that produced that blob. And here it is. So this is now the average MRI signal from that part of the brain over this experiment. And you can see, no math, no nothing, just eyeballing the data, you can see that the signal's higher during the times I was looking at faces than during the times I was looking at objects. You can also see it's higher when I was looking at objects than when I was staring at a dot, something my colleagues love to hassle me about. But the main point here is there's a substantially higher response to faces and objects. Okay, everybody clear on what's shown here? It's pretty simple. Okay, so it's tempting to look at that and say, wow. Oh, and by the way, it's not just my brain. Almost everybody we scan has one of these things in more or less that region. So it's tempting to say, maybe this is a specialized little module in the brain for face recognition. It's our, face recognition is important. We assign a whole piece of brain to doing just that. But these data don't nail that claim yet. They're suggestive, but any one of you can probably think of 5 other accounts of what might be going on there. Maybe this region just responds to anything animate, anything round, anything interesting, and so on and so forth. And it's not until we take seriously all of those other accounts that we're in any position to say, yeah, it's really specific for face recognition. So the whole business of using MRI to follow Gall's research program to figure out if there's specialized bits of brain really requires spending a lot of time testing all those other hypotheses. Okay. So we've been doing that for a while. So here's a summary slide. Now I've switched, and this is the face area again, now on a vertical slice. So this thing on the bottom is the cerebellum. That's a big chunk of brain that lives underneath the main part of the brain up here. And so you can see that this little face area, in me, it's right about in there on the bottom surface of the brain. And so here's some of the images it responds strongly to. And here's some it responds much less strongly to. So raise your hand if you can tell what this image is of right there. Okay, one person plus somebody who knows, 1.5. Okay, raise your hand if you can tell what this image is of right there. Okay, so if you didn't get it, it's a face in profile, eyes, nose, mouth. Okay, this is the same image, it's just upside down. So when I said a moment ago that face recognition just works differently than object recognition, that's based on the fact that it was actually discovered in a PhD thesis in this department in 1969, that the difficulty you have processing upside down faces is greater than the difficulty you have processing upside down anything else. Okay? And so you can see it right here in an extreme case with these images where often you can't even tell that's a face at all. Okay? So that's useful for us because I have all these colleagues who study vision, and they're very serious about the precise characterization of the visual properties of the image. How bright is it, and what is the spatial frequency composition, and all this other fancy stuff. And they want to say, well, your face area isn't really responding to faces, it's responding to one of these other fancy things. And I can say, nope, this is the same stimulus. All I did was tip it upside down, and the response goes down by almost a half. Okay? So that's the kind of enterprise you're in to claim that there's some special part of the brain. Is that more or less clear what's going on here? Okay. So as I said, this bit was fun to see, but not a real surprise. We had all these reasons to think it would be in there. But the next bit we found was a total surprise. And this is a region we call the parahippocampal place area. It's in a piece of the brain called the Parahippocampal Gyrus. And here's another vertical slice through the brain. Again, the cerebellum at the bottom, and it's these two little pink blobs right there. They're roughly kind of straight in about there, also on the bottom of the brain on both sides. And this region, amazingly, responds to images of scenes and places. Anything that has a spatial layout of a place you could be in. Like, pictures of Utah, pictures of houses, even abstract layouts made out of Legos produce about twice the response as objects made out of Legos. And yet, it's not just anything that's large or anything that's visually complicated. We have very similar stimuli. This produces a strong response. You slice it up into pieces, it produces a much lower response when you can no longer perceive the spatial layout. So this region really seems to be specialized for something about perceiving the shape of space around you. We think that probably has to do with the fact that we use the shape of our environment to know where we are when we're navigating around in the world. So the next region we found, our most disreputable, is the extrastriate body area. On me, it's about here, on the outside surface of the brain. And this region, amazingly, responds to all kinds of different pictures of bodies and body parts. So arms, whole bodies, stick figures, silhouettes, all these things produce a very strong response. Even though you can see visually, they're really quite different from each other. And my favorite control is if you take a stick figure of a body and you just move around a few line segments, it's visually very similar, but it's no longer a body, and the response in that region of the brain drops by about a half. So what we have here is 3 regions that respond, each of them very specifically to a single category of stimuli. These things are found in more or less the same place in pretty much every normal subject. We can pop any of you in the scanner, or your kids, as we do, and find these 3 regions in about 15 minutes of scanning. So their very existence raises a whole heap of questions, of which I'll just briefly talk about a few. And then Danny will tell you what he's doing looking at these regions in kids, and Rebecca will tell you about another even cooler region. Okay? All right, so first question, actually, Rebecca, you could pass these out. First question we're going to consider is, let's think about that face and place area. What are those things doing? Are they responding whenever there's a face image on your eyes? Is the face area responding whenever there's a face image on your eyes? Or is it responding when you have a subjective experience of a face? Now, those things are usually the same. Usually, if there's a face image landing on your eyes, you are experiencing a face. But we can use a wonderful trick called Binocular Rivalry, which was discovered in the 1800s, to pull those things apart. So let me show you Binocular Rivalry. If you put on the glasses and you look at this image, first let me tell you what the glasses are doing optically. Close one eye, and you should see just a face or just a house. Okay? Now close the other eye, and you should see the other thing, the opposite image of what you saw before, just a face or just a house. Okay, now open both eyes and just look. You don't need to do anything. Don't try to do anything. Just look and watch. And if anything cool happens, you can say, ooh, ah. Good. Okay, what's happening?
Occasionally the brain wants to pick one image or the other.
Exactly. Exactly. It doesn't want to live with the compromise. It wants to say it's either a face or a house. And so it chooses one for a while, and then it flips, and then it chooses the other. Do most of you see that? If you don't panic. It doesn't mean there's anything wrong with you. It's hard to get the color balance right, so this will work for everyone. The cool thing about Binocular Rivalry is that it's a way for us to change your visual experience without changing what's happening on your retina. Because when it flipped, nothing happened in your eyes. The stuff landing on your eyes was exactly the same. All that happened was a change in your subjective experience. So we can pull these things apart and say, do those regions of the brain reflect what you're experiencing or what's landing in your eyes? Okay? So way back, I did this experiment with Frank Tong, shown here. And we did just that experiment, just like what you guys did here. We taped these little cardboard things on people's foreheads, and we popped them in the scanner, and we gave them a button box, and we scanned them for about 5 minutes at a time, this time here, and they reported with a button box, now I see the face, now I see the house, now I see the face. They would just press different buttons telling us what they were experiencing. Okay? Now, we had found their face areas and their place areas in a separate bunch of scans before, so we knew in each subject exactly where their face area is that likes faces, not houses, and their place area that likes houses, not faces. So then what we can do is look in those areas at what happens during the scan as the stimulus stays the same, but experience varies. Okay? So here is, MRI data are mucky and noisy, and here are some. So here is a several minute scan from one subject's brain. It's probably me, but I'm not sure. Anyway, the signal from the face area is in blue, and the place area is in red. And the letters are the times when the subjects told us, now I see the face, now I see the house, now I see the face. And if you look at it and use your imagination, you might see some trends that, when the subject says they see a face, signal goes up a little bit more. But it's kind of muddy and it's hard to see because there's a lot of noise in these data. So what you can do is you can take all the house-to-face flips and align them all on the button press and average the signal. Okay? And when you do that, the data cleans up a bunch. So here's a house-to-face flip. This is time going this way. 0 is when the subject presses the button. And what you see is when the experience goes from seeing a house to seeing a face, signal in the face area shoots up, the signal in the place area shoots down. Okay? Even though nothing changed on their eyes. Okay? So you may be wondering, why is the change in the brain lagging quite a few seconds after the button press? Anybody have an idea? Remember, I told you the basis of the MRI signal is not directly looking at neural activity. It's looking at blood flow changes that happen after neural activity. It takes a few seconds for those blood flow changes to happen. It takes about 4 or 6 seconds. And so that's the delay here. That's why these peaks are way out here, several seconds after the button press. These peaks are reflecting neural activity that actually happened in time back there when the subjects told us the flip happened. Okay? So you can see here when it flips in the opposite direction, we get the opposite direction flip in the brain. So this is neat. It tells us that these regions aren't just doing something dumb, like responding like automata whenever a certain stimulus lands in the eye. They're actually reflecting what the subject is experiencing. Now, we could push that a little farther and say, okay, if it's reflecting the subject's experience, let's push this to the logical extreme. Let's get rid of the stimulus altogether. Okay, so that's what we did in the next experiment. We told the subjects to close their eyes and just imagine a face or a place with no stimulus there at all. Okay? And we asked what happened in these parts of the brain. So, so here's the experiment. First, this is done with Kathy O'Craven, shown here. So first we do a standard perceptual experiment, alternating periods of faces and places. The subjects saw famous faces and famous to them places. These are MIT buildings. And they just looked at them. So we did a few of those scans. And then we did the same experiment in mental imagery. That is, we told the subjects to close their eyes, and we shouted out to them the names of the same people in the same order, and we told them to just form a vivid mental image of what that person or place looked like. Okay? And again, we scanned their brains as they did this. So what do we see? When we do this, first I'm going to show you a comparison of the response to faces versus places in perception. So here it is now. This is a vertical slice through the brain. So this is this face area that I showed you before, bigger in the right hemisphere than the left, but present in both. So that's the face area. Question is, what does it do in mental imagery? And the answer is, it's still there. It's a little weaker. After all, mental images aren't quite as vivid as actually seeing a face. It's not quite as detailed or as rich, but it's similar, and you can see the very same part of the brain is active. When we look at the opposite contrast, what happens when you see places versus see faces? This is the place area right there, blazing in perception. And here it is in mental imagery, the very same place. Okay? So what this says is that these regions are driven not just by stimuli that come into your eyes. You can also drive them just by thinking, by imagining. And that's good because depending on how you count it, about a third of the brain, pretty much the whole back third of the head, is involved in vision in one way or another. So it's nice to be able to use that machinery not just to see, but also to think visually. Okay, I'm worried I'm going over. Are you watching the time? You wanna, I might skip something. I don't know. Ellen, you want to watch the time? Am I? Okay. Okay, so next question. You may already be wondering, I talked about face, place, and body selective regions. What other bits are in there, right? When will this silliness stop, right? So with a postdoc of mine, we said, well, let's just try more or less everything we can think of here. So we scanned subjects looking at lots of 20 different categories of objects. And this is a kind of unconstrained experiment. How do you know which ones to choose? Well, you don't, so you just wing it. And we chose things for which you could tell a kind of evolutionary story, like spiders are scary, let's try them. Weapons are scary, let's try them. Tools are pretty basic, let's try them. Silly stuff like that. Flowers, that was because there's a line in one of Steve Pinker's books saying that a flower is a veritable microfiche of biologically relevant information. It tells you about the location of future food sources. So, you know, who knows? We could try it. It's an empirical question. We can see if there's a special piece of brain for flowers. So I can summarize the results of this experiment pretty simply. In every subject, we found the face, place, and body selective regions, but we just didn't find selective regions for any of these other categories. Now, with any scientific method, particularly functional MRI, not seeing something doesn't mean it isn't there. It could have been smaller than we could detect with our methods. It could be interleaved spatially with something else so that the signals blur. It could be in one of those parts of the brain that just don't give a very good MRI signal, etc. But at least there are not loads of these. It's not like any old category you look for, you find one as big and robust and consistent as we find for faces, places, and bodies. There are at least a few others that have been found, though I'll tell you about one in a second, unless I should shut up soon. Okay, I'll be all right. And then Rebecca will tell you about another one. Okay, so are there other special bits? Well, there's a couple, but not that we found with that experiment. I think this is my last thing. How do these things get wired up in the brain in development in the first place? How does the brain know to put a face area there, and a body area there, and a place area, well, underneath and in? I wish I knew the answer to that question. We don't yet know. But I'll tell you a couple little glimmers. So first, just thinking about it, genes are important in everything, and you can't wire up a body or a brain without a major role of genes. So it's clear they're going to do something. The question is how specifically do genes specify regions of the brain with this kind of selectivity? Of course, experience is also important. We know that lots of aspects of brain organization are affected by experience. But again, do you only get a face area if you have a lot of experience with faces? Who knows? So this is still a very open, unanswered question, but I'll tell you quickly about two little bits of, actually I'll skip over one. I'll just say, I'll give you one example. This is a guy who did his PhD in my lab and who's now back with a Fulbright visiting. And he did a wonderful study in his lab in China last year where he tested a bunch of twins. He found pairs of twins who are identical and therefore have identical genes, and pairs of twins who are fraternal twins and who share only half their genes. And what he showed is that pairs of twins that are identical are more similar in their face recognition abilities than pairs of twins that are fraternal twins. And that tells you that there's a role of genes in specifying face recognition ability. With other experiments, he showed that it's not just general IQ. In fact, delightfully to me, because I'm bad at face recognition, there's no correlation at all between IQ and face recognition ability. And that's what you'd expect if there's just a whole separate piece of brain that does that. It's a separate process. It has nothing to do with the rest of your intellect. Yeah?
Do good face recognizers have a bigger...
Oh, it's a great question. Wouldn't you think so?
Or is it more
Wouldn't you
irrigated
think so?
with blood vessels or something? Or?
It's a great hypothesis. I would love if that were true. We've been looking at that for a while, and we just haven't seen it. Which, again, doesn't mean it isn't there and we haven't screwed up or something, but it certainly isn't a really obvious effect. So people are now looking at other kinds of less direct things. Well, maybe it's not just bigger. Maybe the size has to do with something like maybe the number of people you know. I mean, it's a silly hypothesis, but it might be true. We're trying to figure out how to test that. But it may be something more subtle about it that determines how well it works, right? Like what exactly are the circuits in there, or exactly how well is it connected to other areas? So this is something we're dying to answer and don't yet have a good answer for. But especially because this experiment tells us that there are very substantial differences in people's face recognition ability. There's something about their brains that are different. And one would guess that this region has something to do with it, but we haven't found exactly what. Okay, I'm going to skip over. There are other fascinating lines of evidence that also implicate genes, specifically in the face area. None of them quite nail it. They're all just suggestive and cool. And I'll just mention one experiment and then stop, and that is to show that experience also is really critical. And one way to look at that is it's hard to raise kids. You guys wouldn't like it if we said, okay, can we borrow your kid for a few years and not let them see faces and see what happens to their face area? That's not cool. But we can look at other things where experience naturally varies. And so we can look at something like reading. Okay, so people have only been reading for a few thousand years. And that suggests that if you have brain specializations for visually recognizing words, they're not likely to be genetically specified. They're likely learned. Okay, so we looked for brain regions for reading. And in fact, we found them. Oops, I skipped way ahead. Okay, so here is a region in the left hemisphere of 3 different subjects. It's tiny. It's about 1/10 the volume of the face area. And here is its response to words and consonant strings up there. Actually, it doesn't care if it's a word or not. It just likes letters. But it responds much less strongly to letters in an unknown orthography, in this case Hebrew. Our subjects didn't read Hebrew. Or Chinese. They also didn't read Chinese. Much less to digit strings and stuff like that. So it responds to letters in people who know how to read those letters. And that's suggestive that this thing must have been wired up by experience. But it doesn't nail it. So to nail it, you need somebody with different experience to show that the response is different. So then we found people who read both English and Hebrew, and everything is the same except the response to Hebrew, which shot up like this. And that shows that the pattern of response in this part of the brain is determined by that subject's experience. If you've learned to read that orthography, this piece of brain responds strongly. So that's at least one piece of evidence of how experience can shape the selectivity of these different parts of the brain. So they're both roles of genes and of experience doing rather specific things in different parts of the brain, but most of this story remains to be understood. And of course, a whole other angle on this is, when do these things develop? And that's one of the questions we're investigating right now that Danny will talk about. And I'll just say there's a huge expanse of fascinating questions that are largely unanswered, like why do some mental functions get their own private piece of real estate in the brain and others apparently don't? What's so special about those ones that do? Like, do we have highly specialized brain regions not only for parts of perception, like the things I've talked about here, but also for aspects of thought? And Rebecca will talk about that. Can these things move over if you have brain damage to one region? Can one of them redevelop someplace else? When we're walking around in the world, we're not just doing one of these things, we're doing a lot of these things at once. How do these things work together to produce our real-world cognition? Which of them are present only in humans and which are present in other primates? And so forth. There's heaps of questions left. So I don't know, do we want to take questions or do we want to move on? What do you think? Okay, go ahead. Yeah, Danny, why don't you come up here and get ready for yours, and I'll take any questions if you guys have them while he's getting set up. Yes?
I have a whole bunch.
Okay, shoot.
Well, have you looked at motion? Because instead of just an object, but like certain motions or flows, or, you know, one thing I'm thinking of, because I was just talking about this recently, I've read a few chapters of the Steven Pinker about how we use language about various physical aspects of how we approach the world. And
yeah, it's great.
Me, that would be very much how
absolutely.
the brain is. Again, like faces and things are programmed into think something coming at you or going away.
Yep, absolutely. So motion is a hugely important visual cue for all kinds of things, right? If I do like that, it's hard not to look, right? So motion attracts attention. Motion carries all kinds of enormously useful information for us. As you said, when things are coming towards you or going away, you can recognize objects by their characteristic motion. If you just stick little lights on them and people walk around in the dark, you can tell if it's a man or a woman walking. You can tell their mood. You can tell all kinds of things just from the motion. And indeed, there is a part of the brain called MT in this country and called V5 in Europe. I don't know why, but anyway, it's somewhere right in there. It's near the body area, right next to it. And it responds more strongly to moving than stationary things. And it seems to be quite specialized for motion. But we're also discovering in our own work, as Danny will mention, we're using movies, which mostly we decided to use. This was Rebecca's idea, because we thought it would be more fun for kids to watch movies than to look at static pictures. So Rebecca and some colleagues of ours made these wonderful movies that Danny will show you. And we discovered, somewhat to our surprise, that the movies just produce much stronger responses than stills. And there's some parts of the brain that respond pretty much only to movies of faces and practically not at all to snapshots of faces. And we think that's pretty cool. And we're actively investigating it right now. So motion matters in all kinds of ways.
I have a few others. There's when you were showing how the going between the house and the face, is any of that related to, this is from many years ago, from school. I know there's something where the neurons, I forget the term, where they get bored.
They adapt, yeah.
They adapt. So is that related to that? And that the changing between the two
yeah,
eyes?
so there are a lot of theories about binocular rivalry and when those things switch and why they switch when they do. And one of the standard theories is that indeed neurons adapt. And you can show adaptation with functional MRI, with other methods. And so it's at least a plausible account that those neurons fire for a while and adapt, and that enables the other neurons representing the other stimulus to take over.
And does that change over time if you stare at something too long?
Absolutely. And you can see that all through the visual system. If you take motion, for example, I wish I had this demo. You don't have it on your computer, do you? It's called the Waterfall Illusion because it was first discovered by, I forget, some vision scientist who was staring at a waterfall for a long time. And he looked away from the waterfall and the whole landscape was going up, right? And the idea there was the neurons that signal motion in this direction were tired out and they're kind of in competition with the ones that signal motion up, leading a net signal up.
Kind of like the various color things you've seen.
Yeah. A lot of color adaptation is in the retina, in the photoreceptors in the retina, but not all of it. There's also some that happens in the cortex and some regions of brain that are involved in processing color as well.
Another question I had, sorry.
Let's take one more and go on, yeah? Okay.
Is the, how much difference is there in the areas between people? I mean, are they within a few percentage of usually the same region or vastly different?
In size, there's quite a variation in size. The position and shape is very stable. So I've been scanning my face area for over a decade, and it hasn't budged as far as I can tell. It's really stable. I mean, I keep saying this. I don't know if it's true. I think if these guys gave me images of 10 different brains with just the activation pattern, I could pick out my brain based on the position and configuration of my face area. So those are there's a lot of it that are very stable individual differences. But this question that this guy asked a moment ago of which aspect of that is related to your face recognition ability, we don't know yet.